All right, well then next up we have Dr. Gabriel Haller from Washington University in St. Louis, and he's going to talk a little bit more about some genetics updates. So whenever you're ready.
Thanks. So I thought I would just talk to you all about the work we've been doing to understand the genetics of Chiari malformation. And the work we've done to collaborate with many of you and hopefully more of you in the future to collect DNA samples from more patients from around the country and even the world so that we can further our genetic studies. Next slide, please. So about a year ago, we started expanding our efforts in collecting DNA, and we collaborated now with the Park-Reeves Chiari and Syringomyelia Center at WashU and the Park-Reeves Syringomyelia Consortium to create the Park-Reeves Chiari Genetics Consortium. And now, particularly for any patients who will see this video now or in the future, we'd love to have anyone with type 1 Chiari malformation contact us and participate in our study. And that can be done as easily as clicking on the link at this website or even scanning this QR code with your phone. Next slide, please. And so what we've done is really expand and make efficient our system of collecting DNA from patients. So what we have is boxes that the patients can just put in one of the blue USPS mailboxes. All the postage is paid for, and no blood draw is required because we collect DNA by simply swishing scope mouthwash in your mouth for 30 seconds and spitting it into a 50 ml conical tube. And that is very stable over time and can be sent to us back at room temperature in one of these boxes. Next slide, please. And so currently we have 11 sites that are all contributing to this consortium, and they're listed here with the PIs. And this was largely, started in large part thanks to the efforts of Dave Limbrick at WashU, who recruited me to come and work in the Department of Neurosurgery to do largely this study, among others. And now we have 4 sites that are enrolling: WashU, Cincinnati Children's Hospital, Vanderbilt, and Nationwide. But these other sites are getting started now. And if any of you are interested in collaborating on this study and contributing samples, you'll all be very welcome. And if you're interested, you can contact Tanda Meehan, who's one of the clinical coordinators with Dave Limbrick, or me directly. And if you're interested. Next slide, please. And so to date, most of our samples have come from our collection efforts at WashU, but a large number have come from Doug Brockmeyer at the University of Utah, Chevis Shannon at Vanderbilt University, partially through the BioVU biorepository there. And we have now started collection at Nationwide and Cincinnati and Vanderbilt directly, and have already collected a good number of samples from patients. And now in total, we've enrolled in our study 1,570 Chiari patients. Now, some of these don't have DNA. About 1,100 have DNA collected, but we have clinical information for a lot of them. And we hope to be able to collect more DNA from those patients in the future. Next slide, yes. And so even just in the past year, we've seen a great increase in the number of patients that we've been able to enroll in our genetics study, and that's listed here, plotted. You can see where certain sites came on board. And very importantly, especially for this talk with Bobby Jones CSF, is that 2 times this year, they very graciously tweeted information about the study on Twitter, and we received massive numbers of participants coming to us online to enroll in the study. So very much thank you for that citation and for helping those patients find us. Next slide, please. And so for all the sites involved, what we do is we send out large boluses of kits. So this is probably 2 of those such groups of kits that we send in big boxes to those sites. And the clinical coordinators work to just take a box, give it to the patient, and the patient can even do most of the consenting online. We have the consent paperwork now, all electronic signatures. And so all the clinical coordinator has to do is explain the study to them and then have them go online to sign. Next slide, please. And for that reason, we actually have patients enrolled in our study from these other sites that are not actually enrolling on their own IRB, but have simply have patients who have come to us through either the Bobby Jones CSF tweets or have found us online by various means and have come and gotten consented by our clinical coordinator over the phone. And then we send kits to their houses and then they send them back through the mail. So some of you may very well be from those sites. And so we may have already started to recruit some of your patients. So there's a large number of patients out there who are very interested in participating in the study. So to date, with the samples that we've collected from patients, we have now over 900 Chiari patients and family members who have been sequenced using exome sequencing. Together with a large group of control patients who do not have, to our knowledge, Chiari malformation, we have performed genetic analyses. Next slide, please. And so that's quickly depicted here. Usually what we do, particularly for rare variant analysis, is comparing the number of rare variants in cases and controls for each gene in the genome. And that provides us a statistical way of determining if a specific gene is associated with Chiari malformation. Next slide, please. And so our analysis that we've now performed on this larger cohort, we actually reported here at this same meeting a couple of years ago, but we have now expanded our cohort to be even larger, and the significant finding here at the gene CHD3 remains very significantly associated with Chiari malformation. So on that plot, just to clarify, on the x-axis is the expected p-value for all of those tests, and the observed p-value distribution on the y-axis. And so further off this line, the dots are, the more significant the p-value. And so usually you look at the top right corner to see which variants or genes in this case are most significantly associated with the trait of interest. Next slide, please. And so that gene is depicted here from N-terminus to C-terminus. You can see all the protein domains, and in blue, the dots represent where the mutations that we see in patients occur. And you can kind of see that compared to the distribution of variants in gray with the red line that are present in large public databases, and compared to the distribution of the black dots on the bottom, which is where pathogenic de novo variants in this gene that cause a syndrome associated with CHD3 that causes macrocephaly and developmental disorders actually occur very much in the middle, in the most highly conserved domains. What we see is the variants largely occur throughout the protein, and that we take that to likely mean that these variants are sort of less deleterious to the protein's function because they don't occur necessarily in the most constrained, evolutionarily conserved regions. And so probably more like milder mutations that cause some of the phenotypes, but not the full-blown developmental disorders associated with the gene. Next slide. And so we actually looked at all CHD genes and observed that there was a large association with a number of these genes, and that overall, if you looked at the number of rare variants in the total class of CHD genes, we saw a very significant association with Chiari malformation risk. Next slide, please. And then lastly, a method that we've used with our cohort is what's referred to as de novo enrichment analysis. And so what we do here is we take DNA from parents who do not have Chiari malformation, or at least to our knowledge do not have Chiari malformation, as they don't have symptomatic Chiari malformation, and a child who has no family history of Chiari malformation, but does have at least 5 millimeters of consular descent. And then we look for variants that occurred in the child, but neither parent. So these are referred to as de novo variants. And when we see de novo variants in multiple individuals with the same disease in the same gene, that gives us very high confidence that those variants are actually causing the disease that is present in the child. Next slide, please. And further, we can look and see if classes of variants are enriched for certain de novo variants. And so what we see here is simply the enrichment over expected of de novo variants in various classes of genes. And what we see is genes that are involved in dominant Mendelian diseases and genes that are highly expressed in the brain are enriched for de novo variants in Chiari patients, suggesting that some of the genes in these lists are actually causative, but we need more samples to identify multiple variants in the same gene in order to prove causation. Next slide, please. But one of the genes that we did in fact see multiple de novo variants in was another gene very closely related to the CHD3 gene called CHD8. These are both chromodomain genes involved in placing down chromatin in the genome. They're highly important for brain development, and both are associated with developmental disorders with macrocephaly as a clinical phenotype. Next slide. And so having found these two associations with Chiari malformation and two genes involved with macrocephaly, we actually started collecting head circumference on all of our patients, and were able to go back in time to prospectively to patients who had head circumferences measured and plot their head circumferences on the expected growth curves that you can find from the Centers for Disease Control. And what we saw was that overall the variants with CHD variants, the patients with CHD variants, and overall Chiari patients in general, both males and females showed increased head circumference compared to CDC growth curves across the age range. And in particular, there was a very significant number of individuals who had above the 95th confidence interval for head size per age and sex, suggesting that these patients actually have non-diagnosed macrocephaly. Next slide, please. And so lastly, what we did was try to, again, model these findings in a model organism. And so we, using this scheme, knocked out, so deleted the CHD8 gene in zebrafish and attempted to look at what effects they had, that deletion of that gene had on brain structure in zebrafish. Next slide. And so we did this using a nice system that exists at WashU to look at fish in mass. So you can throw a plate of fish, so say 96 fish, and do nice Z-stack confocal imaging of the brain in a minute or two per fish. And so this has greatly increased our ability to look at in-depth phenotyping of fish for various phenotypes. Next slide, please. And so what we saw with CHD8 knockout fish was exactly what we predicted based on our findings in humans, is that overall, the brain of the zebrafish here in cyan of the knockout CHD8 fish compared to wild type in magenta was larger. And so, unlike in humans, particularly among people who don't have high quality brain MRIs, we're actually able to show that overall, the brain of the zebrafish as a whole displayed increased size compared to wild type, and it wasn't particularly any brain region. And this was in contrast to overall body length of the zebrafish. And yeah, next slide, please. And so in conclusion, I'd like to first and foremost thank the patients who are willing to contribute their precious DNA to our study and to help us understand the genetic causes of Chiari malformation. I'd like to thank Sam and Betsy Reeves, who made a large contribution to the creation of the Park-Reeves Syringomyelia Foundation, and also to the Chiari Genetics Consortium that we've now created. Enrollment and participation is going very well, and that's, again, in large part thanks to the Bobby Jones Chiari and Syringomyelia Foundation helping us get the word out about our study, and the many great collaborators who are contributing to this study around the country and even in Canada that allow us to collect so many samples in such a short amount of time. And then lastly, yeah, I'd just like to point out that with this amazing patient resource and the genetic information that we've been able to collect on patients, we've identified some of the first genetic factors that we believe to lead to Chiari malformation directly. And we've been able to identify head circumference and potentially brain growth during adolescence or even before in the development of Chiari malformation. So with that, next slide, I'd like to thank everybody who's contributed and the funding sources that we have for these studies, particularly Sam and Betsy Reeves. And next slide, actually I had one more. All the contributors from the sites that have signed on. We wouldn't be able to do this without the amazing collaborators that we have around the country. So thanks.
Thank you, Dr. Haller. Does anyone have any questions? That was really amazing, like all the improvements and advancements you're making in the genetics field. So does anyone have any questions while I get the next slides out?
I have a question. That was a really fantastic talk, Gabe. Fraser Henderson. In 2007, I think it was, Dr. Millerad reported on a very large number of Chiari patients, 13% of which had connective tissue disorders. I was wondering if you had seen in your genetic studies, is there any indication of you finding a gene that might reflect that inclusion, and recognizing that so far they've found no gene for the most common type of Ehlers-Danlos syndrome.
Right. Yeah. So one of the things we found with adolescent idiopathic scoliosis was a lot of connective tissue disease genes, but to date we haven't seen very much signal from genes involved in things like Ehlers-Danlos syndrome, no collagens, other connective tissue disease genes. That being said, I do wonder if we've somehow selected against finding those patients by not collecting DNA, but we're very agnostic with respect to Chiari malformation. Usually, unless you have a known syndrome, generally we'll take DNA. So I think we would find connective tissue disorder genes or genes involved in connective tissue if there was a signal there. But I think we just need more samples. I have no doubt that due to the relationship between those those other traits like Ehlers-Danlos and risk of Chiari malformation, that there is a link there, but I don't know if it's subtle enough to find more common variants that don't cause those syndromes. It's a good question.
Thank you.